friends! Welcome to Science with me. My name is Dr. Erica. I am so excited that you're here with us this week, and our unit is Artsy Science or Sciencey Art. So we're looking at all different things, at colors and movement of images and all sorts of fun things like that. Today we are going to do chromatography. You can do a nature chromatography or an ink chromatography, and I'll show you how to do both of those. To do either of them, what you need is some isopropyl alcohol or rubbing alcohol. You're going to need some plants and leaves if you're doing the nature one. And then you need some permanent markers if you would like to do the other one, along with bowls. You need a coffee filter, and then we are going to hang these with some pencils and tape. So don't need too much for this project. It's really fun. And the way it works is the coffee filter is sort of like, imagine it as like a bunch of different little like play tunnels that things can go through. And if you're an adult and you're really big trying to go through these play tunnels, you got to move really slowly. It's like really hard to get in there. But if you're a kid and those play tunnels are major size, you can zip through those so much faster. And that's the same thing that's happening with these dye molecules that are creating the colors that we see. And so a green leaf might have some green colored molecules, but it also might have some yellows or some browns. And this will separate them out because they're all different sizes and they can run through that coffee filter at different rates. And that helps us separate those colors and see them individually. So if you're doing a nature chromatography, what you're going to do is just pick whatever colorful thing you want in nature. Here's some rosemary. You're going to cut it up into one of your little bowls, and the smaller you cut it, then the faster you'll get some colors out of it. And I sort of chose this rosemary because it looked like it had a little bit of blue in it, which I thought was sort of interesting. And then you're going to add some isopropyl alcohol to that. All right, now it is going to take about a half an hour to an hour for you to start seeing some colors, but you will see it. So this is sort of one of our rhodey leaves, and you can kind of see, if we go this way, that it already has sort of a greenish color in here. And so it's pulling out some of those greens and yellows, but of course, the stuff that we just did isn't quite there yet. So you do need to wait for that to happen. Now, if you want to do an ink chromatography and see sort of what colors is my black Sharpie made up of, what you're going to do is you're going to cut a strip of coffee filter, and you will make a line of black sort of towards the bottom, not at the bottom, because we don't want our marker line to go into the alcohol when we dip it. So I'm going to leave a little bit of space, but then I'm going to make this nice, very thick thing. So I have sort of the bottom here that's going to touch the alcohol, and I can't let the alcohol go up above into the ink because then it'll just sort of spread out instead of going up. And we need it to run through that coffee filter so we can separate the different sizes of dye. And if you want, you can do more than one type of ink. So I have 3 different pens here that we can do. And the nice thing is, for these ones, you can hang all 3 off of the same pencil. So I have an orange. And we can sort of see, what does the orange look like? And I've got a blue pen. Maybe I have a blue pen. Here we go. We have a blue. It's very dark, almost black. All right, so I have these 3 items. And what I'm going to do for this part is I'm going to take a piece of tape, and I'm going to tape them onto my pencil so they're all sort of hanging down. All right, so you don't need huge pieces of tape. I'm going to make them nice and skinny. And I will just tape them straight onto our pencil so they hang down. And then we'll put that pencil into a little jar. And we'll make sure, that it doesn't hang into the liquid. You want to make sure that you're not putting that ink into the liquid. That's really important. If you're doing more than one of these strips, you do want to make sure that they're not going to be touching each other as well. So I take these guys, and now I can just sit them in here and add a little bit of rubbing alcohol, which I actually already have in there, just a little bit, and it's going to slowly, the surface tension is going to pull that rubbing alcohol in, and that's going to dissolve some of the dyes. It's going to pull the dyes in. And the teeny tiny dyes, they can go further faster, so they will end up separating out, which will be really pretty. I'm going to do the same thing for our nature ones. Now the nature ones are not going to be as strong of a visual because we're looking at dyes in nature versus these concentrated dyes that we have in our pens. But you will do the same thing. So you'll take your coffee filter, put it onto your pencil, And then this one, you don't have to worry about how deep it is. You're just gonna plop it in and you're gonna wait for that to come all the way up. And you'll get these really fun, faint lines of color that show us where the dyes and the different types of dyes are in there. This is actually the same way that we look at DNA chunks as we do something called gel electrophoresis. The chunks of DNA at different sizes, the small ones go further and the big ones stay further back. And it's all these things that we can do with science in this thing called chromatography, where we're splitting things apart, seeing how far they can go, and then looking at the colors they make. And we can already see some really interesting things, like this black has some purple dye in it. The orange looks pretty orange, but it is leaving behind some maybe yellowish. And we can keep that running if we want to and see where things go. This one's very interesting. It's still like sort of a dark blackish blue. So I'm sort of curious what will happen with that one. And we can just let that sit as it goes all the way up the coffee filter, and it'll separate out colors. You can do this also with like Crayola washable markers. You could try and see, is it different if I use isopropyl alcohol to soak it in and have it run up the coffee filter, or water, which is another fun little experience. So this is a fun and fast little artsy science or sciencey art type of projects that we can do. Thank you so much for joining us. You can always check us out at patreon.com/rosieresearch. Make sure you subscribe to our YouTube channel so that you know all the fun activities that we're doing as we do them. Have a great afternoon. Bye, friends.